Hello, my name is Margit Olle. I'm from NPO Veggies Cultivation. The title of my today's lecture is Bokashi technology to recycle biowaste into an organic fertilizer. Effective microorganisms. You can see the logo of EM effective microorganism on the left hand side and the bottles of EM, how it is sold, you can see on right hand side. The EM technology was developed at the University of Syracuse in Okinawa, Japan in the early 1980s by Professor Dr. Teruo Higa. EM has been introduced and it is used in more than 140 countries in the world. What is EM? EM is effective microorganism, is produced through a natural process of fermentation, is a combination of beneficially naturally occurring microorganisms, is a liquid concentrate produced from cultivations of a variety varieties of microorganisms. Most outstanding feature is that it includes anaerobic and aerobic species of microorganisms coexisting in a most beneficially productive manner, not chemically synthesized or genetically engineered, contains microorganisms from different groups. EM consists of safe and human-friendly microorganisms, which are main species of EM: lactic acid bacteria, photosynthetic bacteria, yeast, actinomycetes, fermenting fungi. Coexistence. Lactic acid bacteria and yeast are producing organic acid which are used by photosynthetic bacteria. Photosynthetic bacteria are producing vitamins and nucleic acids which are used by lactic acid bacteria and yeast. The concept behind it. In soils, normally there are 5% of harmful microbes, 90% of opportunistic microbes, and 5% of beneficial microbes. And what happens then when you are adding EM in soil? Opportunistic microbes are going to work as beneficial microbes, and in total there are 95% of beneficial microbes, which shifting the balance to suppress diseases. EM technology for agriculture. EM increases yield and improves the quality of production. Organic waste can be converted into fertilizer and EM improves soil. Bioactivity of EM is stable. The basic materials EM1 with molasses and water we get EM activated solution, and EM1 with organic materials you can get EM Bokashi fermented biofertilizer. Cost effective and easy to apply in any place. EMAS EM activated solution. What is EMAS? EMAS is made using EM1 and molasses. EMAS can be used in the same way as EM1. EMAS is much cheaper than EM1. Microbial activity is higher than EM1 if used within 1 month. EM1 liquid type and solid type. Liquid type includes EMAS, EM FPE, EM5, EMRV, and solid type, which includes EM Bokashi aerobic, EM Bokashi anaerobic, EM compost, EM mood ball, etc. EM1 can be activated and multiplied for making EM related materials. Materials: water 90%, and 9 liters. EM1 5% and half liter. Molasses 5% or half liter. Airtight container. Checkpoints for better fermentation. Clean airtight container. Keep in a warm place during fermentation. Ideal temperature is 20 to 35 degrees of Celsius. Check the quality of emas. Fermentation period 7 to 10 days. Depends on temperature. pH should drop to below 4. Ideally around 3.5. Smell. Sweet sour fermentation smell. Unpleasant smell means failure of the process. Color. Dark brown turns into a lighter brown. Gas formation. Generally stops when fermentation is completed. Storage of emas. Keep the container in a dark, cool place. No direct sunlight, container is damaged. Use emas within 1 month. White flakes layer of microbes may form on the surface. These flakes are harmless and do not affect the quality. Differences between EM and emas. EM quality excellent, activity steady, storage 6 months. EMAS quality good, activity high, storage 1 month. Liquid type EMAS, EMRV, EM fermented rice rinse water, and solid type EM Bokashi anaerobic, EM Bokashi aerobic, EM compost, EM mothball. EM Bokashi. Anaerobic EM Bokashi is made in containers and aerobic EM Bokashi is made like here. You can see. The differences between anaerobic and aerobic EM Bokashi. Fermentation conditions in anaerobic EM Bokashi. Anaerobic materials rice bran, wheat bran, rice husk. Fermentation period 2 to 3 weeks. Temperature stable. Used for agriculture as organic fertilizer, animal husbandry, aquaculture as feed additive, kitchen garbage treatment, EM mothballs. Good quality. Aerobic EM bokashi fermentation conditions, aerobic materials, rice bran, wheat bran, rice husk plus fish meal, chicken manure. Fermentation period 1 week, temperature high, used for agriculture as organic fertilizer, others easy to make. EM mothball. EM mothballs can be used for waste water treatment, especially for sludge treatment. It can be reached the bottom and remains longer than emas. Making EM mothballs. Soil ingredients are soil, anaerobic bokashi or rice bran emas. 10 parts of soil and 1 to 2 parts of anaerobic bokashi plus emas. Procedure. Mix bokashi into the soil, pour emas onto the mixture and mix well. Once the mixture has become muddy and thick, form it into tennis ball size balls. Place the balls out of rain and direct sunlight for 5 to 7 days. The balls are ready when they are covered with layer of white fungus. How to apply EM mothball? Use 1 EM mothball per 1 square meter of surface area. Treat once a month until results are seen. It is recommended that emas also be regularly applied together with the EM mothball. You can see here that fishes really like the EM mothball and dynamics row by Mr. Ganit like here. Agriculture. Transplanting. You can use bokashi and you can use emas when transplanting plants. You can add EMAs like 1 to 100, 1 to 500 solution. Growing period, diluted EMAs, dilute 1 to 100 to 500 once a week. Apply EM solution during the vegetation period. Additional menu EM Bokashi 100 grams per square meter or 1 ton per hectare. Beneficial influences of EM. EM promotes germination, flowering, fruiting and ripening in plants. EM improves the physical, chemical and biological environments of the soil and suppress soil-borne pathogens and pests. EM enhances the photosynthetic capacity of crops. EM ensure better germination and plant establishment. EM increase the efficiency of organic matter as a fertilizer. Different brands of EM are applied as follows: inoculation of EM into the soil, spraying of EM on leaves, soaking seeds in EM, EM irrigation, fertigation, insect control. Open land vegetable trials with EM, what I made. The yield tons per hectare of beetroot, swede, Chinese cabbage and white cabbage. With EM and control you can see EM treatment always left hand side and control right hand side. The yield of beetroot increased in EM treatment and in swedes also. The yield of Chinese cabbage and white cabbage were not statistically different. Storage loss of beetroot, sweet Chinese cabbage and white cabbage. Storage loss of beetroot was lower in EM treatment while in sweets was not statistically different. And Chinese cabbage and white cabbage had lower storage loss in EM treatment. Content of calcium. The content of calcium in sweets was higher in EM treatment after harvest. The content of calcium in white cabbage was higher in EM treatment after storage. The content of calcium in Chinese cabbage was higher in EM treatment after harvest. Chemical content. Sugar content in beetroot was higher in EM treatment after storage. The content of nitrates was higher after storage in white cabbage, which is not a good sign, and the content of C vitamin was higher after harvest in EM treatment in Chinese cabbage. The influence of EM on winter garlic yield and its chemical content. The yield of winter garlic was higher in EM treatment, while dry matter and calcium contents were not statistically different results. The influence of EM on summer garlic chemical content. Dry matter was higher in EM treatment compared to control treatment, and the content of calcium in summer garlic dry matter was not statistically different. EM and field pea variety maize. The yield was higher in EM treatment. Magnesium, nitrogen, phosphorus and potassium contents were higher in EM treatment. In summer 2014, 3 New Zealanders from EM organization were visiting us in Estonia, and we went to a farmer right where fields, and you can see here that the soil in EM treatment was almost decomposed, and the soil in control treatment down was not decomposed. And clover root had many side roots and many nodules on roots, while in control only few roots and almost no side roots and no nodules. Fields of right wherever oats treated with 1 liter EM per 230 kilograms seeds, variety ivory, volume weight was in 2015 610 kilograms per cubic meter. Greenhouse vegetable trials with EM. Tomato transplants height and width. Tomato transplants height was lower in EM treatment than in control. The width of tomato transplants was not statistically different. The number of leaves per tomato transplant and the diameter of the stem. The number of leaves per tomato transplant were not statistically different, and the stem diameter was increased in EM treatment compared to control treatment. The content of nutrients of tomato transplants, you can see here a very rare picture. It is unusual that all elements what you are analyzing are giving positive results in a research. So that I found that the content of nitrates, the content of nitrogen, the content of phosphorus, the content of potassium, the content of calcium and the content of magnesium were all higher in EM treatments than in control. Photo of tomato transplants. On the left hand side EM treated plant, on the right hand side control plant. And you can see that on the left hand side you have very fine transplant and it is shorter and with larger stem diameter, and it means that plant nutrients quicker move to the top of the plant and the plant can get through larger stem diameter more nutrients at the same time. Tomato plant from right wherever greenhouse. He used EM ocean salt, biochar, limestone flour, mycorrhiza, variety was titan, sown in the second half of March, transplanted after the end of night frosts in greenhouse, and first yield on 29th of June 2015. Photos of vegetable transplants on the left hand side, EM treated plant on the right, control plant. Cucumber. You can see here that EM treated plant is shorter. You can also see here that EM treated plant stem diameter is larger here in pumpkin and squash. EM effect on the growth of onion leaves. The length of onion leaves was not statistically different, but the number of onion leaves was higher in EM treatment compared to the control at the end of the experiment. Bokashi technology experiments. Introduction. The Bokashi substrate, such as rice bran or wheat bran, is inoculated with a mixture of beneficial microbes that flourish in anaerobic acidic environments. Bokashi fermentation holds several advantages over traditional composting. The process is performed in a closed and anaerobic system. It does not require mechanical intervention, preventing the loss of nutrients and greenhouse gases efflux. The acidic and anaerobic environment quickly suppresses the viability of pathogens. Using the selected microbiome as a starter, the process minimizes the production of putrid odors. This fermentation breaks down the complex organic carbon into short-chain fatty acids, which are readily available nutrients for animals, soil microorganisms, and plants. The prepared EM Bokashi fermentate is thus a compound product containing organic matters at a range of decomposition stages, high biomass of a specific microbial community, and the metabolites. You can see here Bokashi in a container on the left hand side, on the right hand side the grate in the bottom of the container. On the left hand side you can see the food waste in a container, and on the right hand side you can see the top on the container to collect Bokashi tea, which is liquid fertilizer. So on the left hand side you can see the container closer, which provides anaerobic environment, and on the right hand side you can see powerful healthy plants growing on the compost containing Bokashi fermented product. Plant height, the number of leaves and the stem diameter of tomato transplant. You can see here that plant height was higher in Bokashi tea treatment. The number of leaves was not statistically different, and the stem diameter was increased in Bokashi tea treatment. You can see the plants here. Left hand side Bokashi tea treated plant and right hand side control plant, and stem diameter here. Soybean open land trials with EM. Soybean open field trials with EM treated plants. The yield in EM treatment was higher compared to the control treatment, and the yield of soybean plants in 2014 were higher in EM treatment compared to the control treatment. Soybean greenhouse trials with EM. You can see here that EM is improving the germination of soybean seeds. And on the web page www.veggiescultivation.com/books, you can see my 12 research monographs in English and 3 popular scientific books, which of them 2 are in English and 1 of them is in Estonian. Acknowledgements. Thailand Saraburi Farm EM seminar materials, environmentally friendly vegetables production development project results, Right Farm EM experiments collaboration with Kõlleste Garlic Ltd and others materials. Thank you very much for your attention.